The opening slide reads 28th Annual Swartz Foundation Mind-Brain Lecture, Probing the Mysteries of the Mind, AI-Derived Mechanisms of Human Vision. James J. DiCarlo, MD and PhD, Peter Florez Professor, MIT Brain and Cognitive Sciences, and Director, MIT Siegel Family Quest for Intelligence.
Thank you for joining us today, in person and online, and welcome to the 28th Swartz Foundation Mind-Brain Lecture. Mind-Brain Lecture is hosted by the Department of Neurobiology and Behavior and generously supported by the Swartz Foundation.
The speaker is Shoyu Gu, PhD Professor, Department of Neurobiology and Behavior, Stony Brook University.
Established in 1997, the Swartz Foundation has played a pioneering role in advancing theoretical and computational neuroscience. This field brings neuroscience, engineering, mathematics, physics, and AI together to understand how brain and mind work together. The foundation was founded by Dr. Jeremy Schwartz, co-founder of Symbol Technologies and an emeritus trustee of the Stony Brook Foundation. The Swartz Mind-Brain Lecture series was created in 1997 to promote a computational perspective on the brain to and to bring outstanding neuroscientists to the Stony Brook community. It's my great pleasure to introduce this year's distinguished speaker, Dr. James DiCarlo. Dr. DiCarlo is the Peter Florez Professor of Brain and Cognitive Sciences at MIT and Director of the Siegel Family Quest for Intelligence. He received his training at Northwestern University and MD PhD training at Johns Hopkins University and joined MIT in 2002. He served as the chair of the Department of Brain and Cognitive Sciences from 2012 to 2021. Dr. DiCarlo is a leading scientist in neuroscience and a pioneer in integrating experimental and computational approaches to understand vision, intelligence, and the relationship between brains and artificial systems. His work has made fundamental contributions to our understanding of how brain circuits support visual perception. His work has had a profound impact on neuroscience and AI. Today, Dr. DiCarlo will share his groundbreaking research on vision at the intersection of neuroscience and artificial intelligence. Please join me in welcoming 2026 Swartz Mind-Brain Lecturer, Dr. James DiCarlo.
Hello. I'm going to start by saying something kind of provocative, maybe for many of you, maybe not. What I'm going to say is that the phenomena of the mind, that thing on the left, if you will, our behaviors, our perceptions, our sense of free will, all of the things that we call the mind, those things are emergent phenomena of this device on the right called the brain. Now again, that's provocative in some circles, and that's called a materialist stance in philosophy, but it's what most neuroscientists take for granted. I'm going to return to this for the end because I think we're going to start to see a change in how we think about ourselves from the kind of work that I'm going to show you just a bit of today in my own lab, but that I think you're going to see more and more of in our field. So these emergences, this philosophical stance, this materialist stance, if you will, that our mind, everything about us, our sense of free will, how we feel love, all of that can be modeled as an emergent property of a machine called the brain. Again, that's been a stance for some time, but now we can think about actually executing on that stance, to actually start to build such models, and partly test how true that stance is, but also reap the benefits of achieving those kind of models. So I'm going to tell you about some work in that lines today. And AI has been really fueling that endeavor. And I'm going to tell you kind of two stories today to entertain you about what's going on in this area in my own part of the field. So the first story I'm going to tell you is a story about how a subset of human perceptual states can emerge from a set of brain mechanisms and our latest computer models of that. And then I'm going to tell you a story that's really still a science fiction story about how we're starting to think about how these models can allow us to do things that we could not even dream of doing before, that can become the basis of technologies that might someday do things beneficial to us as humans, to modulate mental states in a beneficial way. And again, that's a science fiction story, but I'll give you the conceptual viewpoint on that and show you some work that suggests it's possible in the area that we work in. Before I start on these stories, I'd like to just zoom out a bit and say, we're hearing a lot about AI and its impact on science. And I want you to think about AI and its impact on the field of brain and cognitive sciences. There's one way where you might think about AI, and that's the sense, the first is just as methods and technology as tools. So AI methods and technology as tools is something that applies to many sciences. For example, in our field, things like tools for image analysis or movie analysis allows us to track organisms and to model how flies are moving around or how children are looking, various kind of phenomenologies that we can measure, phenotyping behaviors. That's one example. Of course, at a fine grain, we can do things like on the right here, segment out neurons from other neurons that helps us build up a detailed anatomical called connectome of how our brains are wired together. So these are examples of using AI methods as tools, just like we use microscopes as tools. These are now coming online, have been online for a number of years, and are really accelerating our field. That's not unique to brain and cognitive sciences. It's also just true in sciences in general. And of course, now we have, when many of us think of AI, we're thinking of things like large language models like ChatGPT to help us do things like in science, write our code, or to write our papers, or even write our grants. These are the kind of things that we also can think of as AI as a tool to facilitate the normal activities of science. But what I want to do is shift your mind a little bit today and have you think about AI systems not just as tools to do things like those, as again, those apply to many fields, but think about AI-engineered systems as multi-scale scientific models, as sort of hypotheses of how our brains might work. Tools that help us build multi-scale models that go all the way from neurons and synapses to how they're organized into circuits that then live in different brain regions that then are executing algorithms that underlie what we observe as our behavior and cognition, whether it's about perception or language, how we communicate to each other, or how we move, or how we feel. So that is based on, we know, some multi-stack operations that go all the way down the neurons and synapses and even below that.
The slide adds a second framing labeled AI-engineered systems as multi-scale scientific models, shown as 5 linked stages: neurons and synapses, neuronal circuits, brain regions, states and algorithms, and behavior and cognition.
And AI is now giving us the chance to build models in this space, and I'm going to give you an example of that cross these various levels and the power that comes with that. So this is the first story. This is a story that really I set up my lab at MIT to go after about 23 years ago. And the story was a story of how do we build computer models of how perceptual states can emerge. And this, again, was done with a team of people. Everything I say about this story was really done by these amazing women and men. Some of their names are listed here, but I'm going to highlight a few of them along the way. I'm really just an ambassador of their work. So this first story begins when you think about looking at a scene like this one and you say, what do I see out there in that world? How do I know what's out there? How do I know there's cars or people or signs? Those are called objects. How do we recognize the content of this image? This image is really just a whole bunch of pixels coming at you, yet we can extract what's called the latent content. That's a problem that's been long fascinating to computer scientists and to neuroscientists like myself. And one of the things that we knew when we studied this problem is that when you and I look at a scene like this, we don't just take it all in and know everything at once.
On the slide, a red zigzag line is drawn over the street scene, connecting several points across the image: a person, a car, a sign, to illustrate one possible scan path with about 200 milliseconds at each point.
We actually move our eyes around in a scan path that might look like this one, where we dwell for a little bit of time, for about 200 milliseconds, just the blink of an eye, literally, at each location that we decide is interesting to us. And that's when we see, that's when we estimate content, and then we move around and see other parts of the scene. So we can now think of just what's happening in just one glimpse of the scene and just a little bit of the visual field that I've illustrated here for just a 200 millisecond viewing. And we're taking many samples of that as we go around the scene as I showed you. So this reduced the problem to a problem we call core visual perception, the capabilities to be able to say what's in just that little bit of window there in just a short viewing time. That's the problem we focused on. And that turned out to be fortuitous. It allowed us to develop models that explain this phenomena and also many other related phenomena. So if I want to just fix ideas here, we're just thinking about, can you estimate in 200 milliseconds that that's a car, that there's a person in that scene, that there's a sign in this scene to the right. So that's an example. Of course, we can also have estimates of what's the direction of the person or the direction of the car and other various variables that we can measure in this space. But just to fix the idea, think about the primary noun or object in the scene. We studied that not just in humans like you and I, of course, shown on the left, but in our evolutionary cousins, the rhesus monkey here, shown in the middle, an example of a rhesus monkey doing a task like we would ask humans to do, which is to click a screen, see an image again for just a few hundred milliseconds, and then make a choice as to what the organism thinks he or she saw. In this case, by clicking the right or left button. And you can see this non-human primate doing that. Now, in data I won't show you today, we showed that monkeys and humans are very, very similar in their abilities to do this. Not just that we can do it, but that when a mistake is made by a human, it's also typically made by a monkey and vice versa. So we see the world in the same way. In fact, again, we're evolutionary cousins, and so it makes sense that we make the same kinds of mistakes and the same kinds of successes. So we can think of us as sort of biological twins, and that makes this a very good animal model that if we can understand the processing in the rhesus monkey, then we will directly begin to understand the processing in the human model. So our lab set about studying that process in the rhesus monkey because we have tools that we can use that are more invasive and allow us to study the neural hardware at a finer grain level than we can do in humans. But again, the goal is always to understand human visual processing. So there's a monkey brain on the right and a human brain on the left. And neuroanatomy from the monkey studies decades before us provided us a kind of a roadmap as to what was going on there.
The slide shows a complex wiring diagram with about 25 labeled boxes representing cortical areas, connected by many crossing lines, arranged roughly left to right from the retina and LGN through the visual cortex.
This is an example of all the visual areas in the monkey brain that had been mapped out again, by decades of neuroanatomists, showing the connections between the major areas. Each of these is like a little postage-sized area of the brain, and they connect to each other in what's called a hierarchical pattern from left to right. The eyes capture the data from the world, of course, and they travel through an area called the LGN and the thalamus, and then connect into this network of regions. These are, again, representing millions of neurons in each different area.
On the diagram, a row of areas at the bottom is now highlighted in yellow as the ventral visual stream. On the brain image at upper right, the regions V1, V2, V4, and IT are shaded to show the same pathway.
And we're especially interested in this connected set of areas at the bottom, now highlighted in color, called the ventral visual stream. And that's now also shown in the monkey brain above.
The slide adds a pipeline below the brain diagram, showing a car image entering at the left and passing through labeled stages, RGC, LGN, V1, V2, V4, and IT. Each stage shows a small grid of colored dots representing neural activity patterns at that level.
And the reason that we're interested in the ventral visual stream is that we know it's involved in recognition. We knew that lesions in that area produce deficits. So the story was we had this set of connected areas, but we wanted to understand functionally what was happening in those areas to allow it to support recognition tasks like the one I showed you at the beginning. One of the first things we did was to show that neurons at the top of the ventral stream, this area called IT here, which stands for infratemporal cortex, could support the linear readout of what was in the scene. Was it a car? Was it a person? Was it a sign, as I showed you earlier? Whereas the earlier areas could not do that. So this was evidence that these areas support it, along with all the other evidence from the field. But what we really wanted to know is what are the computations happening along the ventral stream to get to IT cortex?
2 photographs are added showing electrode arrays placed on the monkey's brain at 2 recording sites. The pipeline now ends with a label reading Core Object Recognition, Behavior.
And so to do that, we and other physiologists record from neural activity in the ventral stream. So now I'm going to animate this picture. And instead of thinking about just a static set of anatomical areas, We're watching these areas function online when images like these are flashed to an animal or to a human. The same processing we think is going on. Just like you're watching this right now, your brains are producing these neural patterns inside your head at these different levels of visual processing. And so now we can record those areas and start to try to infer what are the computations that are happening. And you're going to see this drawing at the bottom a lot. That's, to me, that's representing for you a set of brain areas that we know support this task that we're trying to model with machines, computer models of what's happening in the ventral stream. So to do that, to understand visual perception, we needed to build these computer models of those mechanisms. And I already showed you the rough neuroanatomy.
The equation on the slide reads R of t equals the sum over I of w sub I times u sub I of t, meaning the neuron's output is a weighted sum of its inputs. Below it, if that sum is greater than 0, the output is the sum itself. Otherwise, the output is 0.
So what we need to do is to start to build, we thought, neural networks that could approximate the biological neural networks that lived in there. And neural networks are essentially ideas of where you have a neuron like this one, a simulated neuron, shown here as a simple point neuron, which sums inputs from other neurons, weighted by something that we think is akin to the synaptic strength of how one neuron connects to the other. And then that neuron projects further out to other neurons downstream. So this is just one neuron in a possible neural network. And there's some nonlinear function that describes how the weighted sum is then translated into the output here, shown as R parentheses T, output varying over time. This is the core element of an artificial neural network, and it's meant to approximate a biological neuron, here shown at the top. Of course, one neuron doesn't do that much. It's populations of neurons working together in a network that does interesting things. So we wanted to model the populations of neurons that live in the ventral stream. So think about a bag of neurons like this that are somehow connected to each other with some numbers that we're going to have to determine.
On the slide, an empty box appears labeled model of one cortical area.
That would model one of those postage size areas that I showed you. And then maybe we don't just have one postage size area, but we have a set of them.
The slide adds a row of 4 empty connected boxes, representing a hypothetical pipeline. Sensory inputs flow in from the left, with forward arrows and feedback arrows between stages. The boxes are unlabeled at this point.
Here's those 4 major ones I showed you, V1, V2, V4, IT, and they're connected to each other, and they receive sensory input from the eyes, and they're connected to each other in, say, a feed-forward pattern. There's also feedback connections that we would like to model. And there's parameters that specify all this. So when you design this in a computer, there's a whole bunch of numbers called parameters that specify how many neurons you have and how are they likely to be connected to each other.
2 labels appear on the slide: parameters to specify connectivity strengths under the neuron model on the left, and parameters to specify architecture under the pipeline boxes on the right.
Many of these parameters are in these so-called weights, the connections of one set of neurons to another, shown here on the left. And in fact, models of the type I'm going to show you have millions or even billions of parameters in them. And we're going to return to that in a moment. So this was the textbook description for a number of decades of how we're able to see the world, to see object content in the world, is a stacked set of visual processors that are cortical processors that process the visual information to give rise to our percepts of things like objects. Again, this is what the textbooks would say.
The slide is now split into 2 panels. The top, labeled Conceptual Model, shows the row of empty connected boxes. Below it, a green-bordered Brain panel shows a pipeline starting with a car image, passing through stages RGC, LGN, V1, V2, V4, and IT with a brain inset at lower left.
That's a conceptual model, though it isn't actually yet an implemented model, but it was meant to somehow approximate the biology that, again, I'm showing you here at the bottom. And so now those networks, when you build them and add in other constraints that we know from the physiology, they're known as deep convolutional neural networks. So they're not arbitrarily connected, but they have structure within them. And they're usually drawn in engineering books in a picture that looks more like this at the top. So I'm putting this here, but telling you where those families of models came from. So they're a set of linear, nonlinear operators stacked up in a deep network, in this case, 4 levels deep. And the villain in this story that I'm trying to tell you is the one I've already referred to, which is we already knew as neuroscientists, this is the neural network. It's somehow important to vision, but we didn't yet know how to set the parameters, the millions or perhaps billions of connections between those neurons. We didn't know how to measure them directly. As I mentioned earlier, there's efforts to try to get into the anatomy, fine-grained, and measure the connectome and try to get that. It's still very hard to do that. How are we going to estimate all these numbers from making recordings from animal brains or measuring behavior? How are we going to do that? And the big breakthrough came in around 2013, where some AI-forward methods, essentially machine learning methods, helped save the day from this story. In my group, this was led by a postdoc, Dan Yeamans, who's a professor at Stanford now. And what we did was use optimization techniques to tune all those parameters, to essentially search the very large base of parameters to find sets of parameters that helped the network perform tasks like visual object recognition. In a sense, we were training the network. We were being early AI engineers to say, let's take this kind of network, teach it to do tasks like vision, and then see if that helps get the parameters in a space that makes sense with respect to the biology. And that was something we weren't able to do before the advent of large data sets, large image sets, the techniques to optimize, the computers to do it, and all of that came together. And for us in 2013, we started to build those models.
The slide title changes from conceptual model to implemented model. A series of assorted images appear, feeding into both the artificial model on top and the brain pipeline on the bottom as the shared input image.
So now what's great about this is we don't just have a conceptual model, we have an actual computer model that, like our brains, can take images and make a prediction of what should be happening at each of the levels of this system. So the neural network at the top is making predictions of how its neurons should respond to these images, and the neural network at the bottom, the monkey's neural network, is also actually responding, and can make those measurements.
A compare arrow appears between the artificial network and the brain pipeline at the IT stage.
So now we can do the really interesting thing of comparing, do the neurons at the top in the artificial network, are they actually predicting the neurons at the bottom? And so we can take a neuron out of the biological network here that we're measuring with an electrode nearby, compare it with the responses of a neuron inside the artificial network. Now there's a little bit of technique here. We need to find a neuron in the artificial network that aligns with a biological neuron because the network doesn't provide an exact detailed map. And there are a number of techniques to do that, but in short, they amount to first testing with a little bit of data to find the nearest twin, if you will, the digital twin that's closest in the artificial network to the neuron that we're observing, and then test with some new data. Are we able to predict the neuron that we're recording? And this is just showing you to fix an idea for one example neuron. And we did that in IT, we did that in V4, and colleagues of mine did this in other areas, V1 and V2. So we're able to make these fine-grained predictions at the single neuron level of this big network with lots of parameters with the actual neurons in the brain.
Two images appear at the right side of the slide. A biological neuron image at the end of the brain pipeline, and a human face representing the perceptual report side of the comparison.
We can do this not just at the neural level, but also at the perceptual level where it's straightforward to take the monkey or the human reports, as I said, are very correlated, almost identical, the mistakes that the monkey and the human make, and see if they line up with the mistakes that the network makes and the successes that those networks make. So we can make those comparisons too. And the really, really exciting thing for us is that these networks started to align with the neural data far better than any models that we had before this. Again, this combination of techniques led to networks that could do that.
A New York Times article appears, headlined, "Scientists See Promise in Deep Learning Programs."
And I'm going to give you a sense of what we can do with this. I want to make an aside here. At the time that we were doing this, this was also when computer vision was starting to rediscover that these kind of biological networks were actually quite good. In fact, in 2012, this is from the New York Times, they were saying, we're starting to see that our AI does better if we build these neural networks. And it's inspired by brains and inspired in exactly the kind of data that I showed you, how to recognize patterns and start to use it for other technological goals, not just computer vision, but a whole range of other applications. And in fact, that was the early precursor of this current wave of AI to optimize neural networks with large data sets to do lots of things beyond vision, and including things like now upgraded versions of these to do things like the large language models. Okay, that moved well beyond the vision roots that it was inspired by, but this surge in work was happening at almost the exact same time that we were making these comparisons with brains, with us trying to do these kind of optimizations. I want to turn back to the biology and less so the tech side to say, what does this mean for us to understand the biological network? So again, around 2013, we were doing this, and here was a series of papers we started to produce about this.
A chart on the slide shows number of publications per year for the search Artificial Neural Network and Human and Vision from 1988 to 2025. The bars stay near zero until around 2013, then rise sharply through 2025.
And in fact, the whole field in vision now tends to model the ventral stream and other visual areas by using these same kind of performance optimization approaches. This is the number of publications. Nobody was using much neural networks for a while. And then as soon as this work started happening, we were able to show this approach could produce the leading models. Now it becomes standard in the field to use this to kind of find these hypotheses, find these models aligned by doing these kind of optimization approaches. And that, of course, will continue. These models are not yet perfect, but they're far better, as I said, than models we had before. So let me tell you that this story still continues. So this story of how we wanted to build models, we had an idea, we had to use AI-forward methods to find the better models, and we did find things that were way better than things we had before, but again, still not perfect. And even today continues to find more and more brain-aligned models by more and more techniques in this space. So and sometimes you can think of this as better and better efforts to build digital twins of the ventral visual stream. We and others track this on an open science platform that we're part of called BrainScore. If you're interested in seeing where the latest models sit and how well their predictions are doing, this is really not one lab's work. It's really a community set of efforts to try to build models in this space. And we're just one player in that larger goal of humankind to understand, in this case, visual processing.
The slide shows 3 images stacked vertically. A frog photo labeled frog, a noisy gray pattern, and a third frog photo that looks nearly identical to the first but is labeled not frog, error. The slide illustrates that adding the noise pattern to the original produces an image that fools the model.
I want to sort of give you an example of how the models have actually advanced a little bit, and this will also introduce the second story to a way. So one of the problems of these models that was identified early on, even though we showed they were good brain alignment, they had this weird behavior that if you could take an image like this one, the model might say, it's a frog, but you could do something called an adversarial attack. So you could attack the model and use the fact that you had full access to the model to ask it to try to fool the model, design a little additive image, which you can barely see here, but there's a little bit of pixel, like not noise, but structure in this image, added to this to produce this image here. And you do that in a targeted way where you're attacking this model to fool it, to think that this is not a frog anymore. And it might, say it's a cow or it's a church even. So just by doing this, you can fool these early models in this way. Yet you and I look at these images and we can barely tell the difference, which implied to the biologists like me that this means this model is not perfect. Again, there's something wrong with it because this is an image that none of us would call not a frog. We wouldn't call it a church, for example, yet this model was doing that at the time. So this, to some people, suggested these models have problems, and of course, no models are perfect. And here's a quantification of that. Here's an example of like attacking a model with bigger and bigger size image pixel manipulation. And on the y-axis is how many mistakes the model or the human makes. And this is 100% mistakes up here. And this is just showing you quantifying when you move, kind of allowed to move enough pixels around intuitively that in a targeted way, that can lead to big disruption in the model's output. Yet humans are barely affected by this, just like you're looking at these images now and barely noticing the difference. So, but that was the vanilla early yesterday's models.
The slide shows 3 side-by-side charts. All share the same x-axis, perturbation pixel budget, how many pixels are changed, and y-axis, percent errors, from 0 to 100. Left panel, no model. Both humans and the model stay flat near 0. Middle panel, yesterday's model. The model's errors shoot up to 100%, while humans stay flat. A huge gap. Right panel, the new model. The model and humans now track closely together with almost no gap.
More recent models have now come along, and they are able to show that when you make these same kind of attacks, when we start to affect the model's output to disrupt the model to make it think the image has changed to something else, which you might call an error relative to where it started, the humans also tend to change their percept, or in some sense you could call it make an error, or at least change their perceptual report with respect to the change in the image. And that's what we're showing here. And humans and models are very similar now. And there's still a gap, but it's not much of a gap, almost no gap left. And again, more work to be done, but just this gives you an example of how the model started off really interesting and just continue to get more and more human aligned. And this is one example. To give you an intuition of that, I'll show you on the next slide here.
The slide shows a grid of dog images arranged around a central image labeled dog. Each surrounding image looks like a dog but is labeled with a different category: insect, cat, crab, turtle, bird, frog, primate, fish, illustrating that small perturbations can shift the model's perception in many different directions.
Here's an example of starting with an image of a dog and asking just the model to move a few images to make the model think it's now a crab, for instance here. And I think when you and I look at this image, even though very few pixels have been changed, you might, if you just looked at this to begin with, you might have never even guessed a dog. You might have said a crab, or a bird, or a fish, or a primate, or a frog. So you can, nearby any image, find a little perturbation that the model helps us search through that affects both us and the model output. And again, that suggests that we're aligned with the model and also is an interesting tool for doing things like graphics and other things here. Okay, the big picture that I want you to take from this, though, is probably best illustrated on this slide.
The slide shows an empty plot titled A Big Picture, Y-axis brain score, X-axis performance on visual object recognition tasks.
And this is the takeaway from this thing that I told you about vision that I think applies to other parts of our field. So here what I'm going to plot for you is an example of performance on a task like we might train a model to do. Like you might think of this what an AI engineer would do. In this case, tell me if there's a cat or a dog in the image. I'm going to train it to do a visual object recognition task. In this case, over a large set of objects, thousands of object categories. And on the y-axis, I'm going to plot the alignment of the model with all those neural data that I was referring to earlier, what I called the brain score, which is going to align how much neural alignment is there, how much behavioral alignment is there. It's an amalgam score of all of that. And this is for the area of visual models trying to model the ventral stream that I just referred you to.
The plot shows a cloud of dots rising from lower left to upper right, with a correlation of R equals 0.92. Dots labeled 2013 sit near the lower left of the cloud. Dots labeled 2018 sit higher up the trend.
And here's the data that we and others started to produce, and this plot continues today. Each of these dots is a new model with a new set of parameters and perhaps even a new architecture. And what you can see here is that there's a trend as if the sort of AI engineering gets better, the brain alignment at all these neural levels is also getting better. And this started again around for us in 2013. We showed this trend, and it was a big part of what we inspired people about this approach. And then this continued while computer vision was, in fact, adopting these techniques, and they were the ones building the models, not the neuroscientists. But we as neuroscientists were showing that they were actually aligning better and better with the brains, even though we weren't the ones building those models. Again, AI engineers or computer vision scientists were building them. This trend has now since flattened out, and that's an interesting side story, but I hope you can get the general idea that our field has pushed into a new regime because of this here. This is not just true for vision, it's also true in things like audition. This is my colleague Josh McDermott and some work from his work in his lab at MIT. Here is performance on an acoustic word recognition task, and this is alignment with brain data collected from humans in fMRI. So it's a different scale of data, but it's still a neural measurement, and the same trend applies. That is, you make models better, you tend to better align with the brain data. Here's my colleague Ev Fedorenko's work in language areas of the human brain, areas that are involved in processing human language. And here are large language models now, the precursors of large language models shown on the right. And as each of these dots is a different large language model, and they've continued off to today. But again, the same trend is as you make them better, as you do better AI engineering, you get better alignment with the neural responses, in this case in humans processing language. So the big picture story here is that in each domain, as we do more AI engineering in a space that's at least roughly brain constrained, turns out to give us better neuroscientific models of what we'd call the mechanisms that are operating inside the human brain, whether it's vision or audition or language. And that story offers a kind of an approach that we can continue today.
New slide title, A Research Strategy. On the left, a natural sciences icon labeled Human Intelligence. On the right, an engineering sciences icon labeled Artificial Intelligence.
It says we need to embrace the interface of natural sciences and AI engineering. The way I think about that is areas of natural science, like studying human intelligence, whether it's neuroscientists or cognitive scientists, producing discoveries, measurements. For example, think about the anatomy that I showed you that led us to think about that class of models. And then an engineer is doing theory and building, creating ideas of how to actually constrain those models, to optimize the parameters of those models, coming together to build the artifacts, which are those models, the integrated machine executable models of aspects of intelligence, in this case, part of core visual intelligence are the ones I've been telling you about. Those actually turn out to be then our leading hypotheses of the mechanisms of intelligence, just like I showed you in the last slide for vision. They also serve as technology drivers, as I showed you. The field of AI sort of fed off of this for a number of years and continues to evolve even past that. But this loop continues today to execute this, to further refine the experiments and measurements based on now these more leading models of what's going on in these brain systems. And theorists also take these models and say, you know, we can make more distilled versions of them, more theoretically grounded versions of them, more interpretable versions of that, and that work continues as well. And all of this feeds back to better models that inform both of these areas, both artificial intelligence and human intelligence or natural intelligence. So this virtuous loop, we think, is building a new science of intelligence, broadly speaking. And wearing my hat as director of the Siegel Family Quest for Intelligence at MIT, that's something that we are trying to execute on at MIT and with partners outside as well.
This is a return to a previous slide labeled AI-engineered systems as multi-scale scientific models, shown as 5 linked stages: neurons and synapses, neuronal circuits, brain regions, states and algorithms, and behavior and cognition. The slide adds a domain vision label on the right of the pipeline.
So let me zoom out a little bit. I've been telling you a story about how AI-engineered systems can give us multi-scale models. Those models that I described, they span the scale all the way from neurons to being neurons organized into circuits that live in brain regions. I showed you 4 examples that are executing algorithms that are effectively approximating our behavior, in this case, object recognition behavior. Told you a story of how our models of that, where they came from and how they're continuing to evolve. So now we have these kind of computer models. I showed you that for the domain of vision, and then I should have alluded to the fact that this is also happening in audition and also in language, and I think it's going to continue in other areas as well and grow into other areas as well. That was the first story I wanted to tell you, that we can take this idea seriously, that we are machines and that we can build models of those machines, at least parts of our function, in this case focusing on vision. But now I sort of want to turn to this sort of beginning of the second story, which is, why should anybody care? Here's this model of the visual system. Of course, scientists care because that's our job, to build models or to try to understand the natural world that we're studying. But why should we care? And here I have a bit of a personal crisis that happened to me. So I used to stand up and give talks like this, now starting almost 10 years ago, on this kind of work. Again, this is a story over a decade. And one of the questions I would often get at the end as I present these models and say, wow, we've succeeded in modeling these neurons and we've accurately predicted their responses at multiple levels. Areas that used to be mysterious we're now explaining with these models. And the question I would often get is in 2 forms. It's like, well, Jim, you know, you really haven't understood the system. You've basically converted the biological monkey into an artificial, complicated system. So a complicated biological system transformed into a complicated artificial system. That's not really science. You just sort of moved it into a computer. It's like you've copied the monkey into a computer, but we don't have intuition of what's going on. We don't have understanding. And, you know, some others would say, well, you know, you don't have Maxwell's equations or something very clean like that you can hand us. You have this big, messy artificial network with a bunch of parameters. And, you know, for a while, I was like, wow, I thought I'd done a good job here. This is, you know, I worked hard, and then it's not satisfying yet to people. So I thought about it for a while. I'm like, you know, there's a point here. We need to be able to say, either we need to have something that's cleaner to intuit, or we need to show that having these even complicated, hard to intuit models is useful. So as an engineer, I took the second approach, and I'm not trying to downgrade the first approach, the sort of theoretical cleanliness approach. It's just I'm not a theorist by training. I'm more of an engineer. So I said, you know, let's take the applied approach. What can we do with these models that we couldn't have done before these models? What, if anything, can we do? And so one of the first things we decided to do was to explore something that we call neural control or neural modulation. And so I'm going to tell you a bit of that story, and that's going to lead into the science fiction part of this. this.
On screen, the 2019 science paper by Bashevan, Carr, and DiCarlo, titled Neural Population Control via Deep Image Synthesis.
The idea is that, okay, and this is Priya Bashevan, who's now at McGill, who was a postdoc in my group, and Cohidis Carr, who was also a postdoc, and they worked together on this. He's now runs his own lab at York University. And what we did together was we said, could we do something which we call inverting the model?
The slide shows a closed-loop diagram. A cat image enters from the left through an eye into a deep convolutional network with stacked layers ending at cat, then connects via numbered arrows to a monkey brain on the right with regions labeled CIT, AIT, PIT, and finally back through a gray screen and another eye on the lower left.
So here's an example of one of these early models. So it's modeling this set of areas. Here we're showing a few of them. This is this middle area, visual area V4. So it's like halfway up the processing chain. And this is one of these early models that's sort of trying to model V4 and like as this third level of this model. This is, again, one of these deep convolutional networks. Remember, it can take images in, and it processes them, and it tries to align the neurons in here with the neurons that we're recording in the brain. And we can show the monkey and the model the same image here, which is, in this case, this cat, for example. But now once we have this aligned model, we can do this kind of neural control idea. And the idea goes like this.
A title appears, an application goal. Set a desired neural state of a target brain region.
You sort of say, I really want this particular V4 neuron to be really active and maybe all its friends to be inactive. Or I want every other neuron to be active and all the even neurons that are recorded to be active and the odd ones to be inactive. Whatever your control goal is, you can pick any goal like that you want. You can then ask the model, model, could you please find for us an image that moves the neural state into that regime? This is a kind of an interesting scientific thing, but you can also see it potentially has applications, which I'll refer to in a moment. And so you work back through the model, you tune the pixels under the guidance of the model to try to make that happen. That's what's illustrated in this red line here. And to show you kind of this is kind of one of our early work on this.
On the slide, two side-by-side photos appear of what looks like the same shaggy white dog, visually nearly identical. Between them, an arrow indicates a small image change predicted by the model.
We would basically start with some noise image, and then we would optimize until it got to one of the control goals that we had, and that's what you see happening here at the top is another piece of computer technology using this model to try to, as a guide, to help it find an image that it's going to set the neuron that we're after into a high-level activity state, for example. I don't want you to think about the details of the control state. They don't much matter here, but just the conceptual idea that you can invert the model to design the stimuli. And so you can think of this as designing interventions, precise patterns of photons that are going to strike your epithelium, in this case the eyes, that are going to somehow then be transmitted into the brain to produce an activity state. In some sense, that's what the model was trying to predict. And now we're doing it in a kind of a control loop rather than just present images and just observe, which is what I showed you earlier. We're designing to control the neurons. And we showed this actually worked quite well. This is in 2019 for this visual area. Here we were just trying to go from like white noise patterns to just some great activating image. And you see it looks like some set of bars. Doesn't much matter, but it was just trying to get the neurons activated. And this set the conceptual frame for us, and we tested in the monkey like that. This led us, though, to think about, well, what can we do beyond as a kind of scientific test or kind of a parlor trick, if you will, that we could do here with the model? What other things could we do that are richer than that in an application space along this line? And so here I want to begin to tell you in the last part of the talk here about this science fiction part 2 story. So this is a possible crazy science fiction story. I only have a little bit of data to kind of add to this, and it's more motivational than actual reality yet. But I wanted to tell it to you to give you a sense of where we're going and where we're trying to test next. So the idea begins like this. Like, what if it were possible to modulate the activity in some specific set of neurons deep in the human brain in some beneficial way? I'm going to say more what I mean by beneficial in a moment as ideas, but imagine that was possible. Okay, sounds good so far. What if we could do it without giving you any drugs? And what if we could do it without any electrodes or other devices stuck into your head to make that happen? Instead, what if we could do it by simply modulating the pattern of light striking your eyes, maybe without even disrupting your ongoing natural perception, and with you still having control over whether you want this to be happening to you or not, without bypassing your free will. Some level, this is like maybe it's the beginning of, I don't know, some science fiction movie. It does sound like a little bit of science fiction. How am I going to inject these specific patterns deep into your brain without anything injected into your head, and to do it in this way that almost feels like magic? So this is science fiction, but we think there's a chance that some aspects of this could be science reality. And let me sort of give you a sense of why we think that at the moment. Okay? The idea starts with the discovery driven by these control experiments, which we extended deeper into the brain, into this area IT, which is one level past V4. So let me just show you an example of one of the sort of surprising things we found. So imagine we're recording from a neuron inside a monkey area IT, deep in the visual system. And our computer models started to tell us, look, if you attack these neurons using the same methods that we were using that I showed you earlier, where people were using to attack the computer models, and I showed you how that had improved, you now turn those methods to attack the neuron. In some sense, you ask the model to find image changes that will make the neuron really change its state from, for instance, low to high. And you could tune it for any neuron that we're actually recording that's been mapped into a model. And what we found is that the model would tell us not only is it possible, but when we tried those out on the actual neuron, that most of the time the model had successfully predicted the attack would succeed, that it did succeed. And by attack, which is a word I don't really love, it came from computer vision, because I want to think about beneficial applications, but let's just call it modulate the neuron in a predicted way.
On the slide, two side-by-side photos appear of what looks like the same shaggy white dog, visually nearly identical. Between them, an arrow indicates a small image change predicted by the model.
So here's an example of this one example neuron where we recorded for one image. We said, model, we're going to show this neuron this image. It has a low response to this image, but I'd want you to modulate this image slightly if you can and make it have a very high response. The model returned to us and said, please change the image from this to this, and that will dramatically change the response of this neuron. It's what the model predicted would happen. And what I'm going to show you in the next slide is it actually did happen. And so it's not just a random change, it's a very specific model-guided change that we would have never been able to find without the model. So this is just to give you an intuition of what's possible.
The slide now shows two labeled rows. A preferred category row at the top with images of valves and a high firing rate raster plot, and a non-preferred category row below with images of dogs and a low firing rate raster plot. X-axis is time in milliseconds, 0 to 300.
Here's actually some data. This is like recording data from a monkey brain. Recording now, these are like raster plots. Each of these lines is an action potential from a neuron, which is how neurons talk to each other. Lots of tick marks indicate high rate, like here. Little tick marks equals low rate. So you see low rate here in this blue window, high firing rate here in this window. This is the normal time scale in that blink of an eye, a few hundred milliseconds, that these neurons typically respond. These images, we call them clean. They're just filled out of a database, so we call them clean images. And this is an example of a neuron responding very low to these images of dogs, including the one I just showed you, and responding high to these images of valves. In fact, this response firing difference between these two neurons and all its friends is typically what we thought would give rise to the animal's ability to say, for instance, this is a dog more than a valve, or vice versa. And so that's a standard thing that we would have seen in textbooks for 20 years, ahead of what I'm going to show you next. The surprising thing was the model led us to this.
A third row appears below, labeled epsilon equals 2.5. Same dog images, but now slightly perturbed. And the raster plot shows dense high-rate firing, matching the valve images at the top.
Which is to say, nearby this image, there's a slight perturbation, and I just showed it to you, of a very small magnitude, which is quantified here, that it predicted would produce a very large change in the neural response. And here's the actual data from this neuron recorded, and you can see now the neuron's going crazy to this image, whereas it didn't like this other image. So just nearby, we can make a subtle change in the image, which dramatically changes the firing rate of the neuron. And I showed you this up here so you can contextualize. This is a large change relative to what we typically think of as the operating range of this neuron, which is illustrated here. So this was quite surprising to us that the model predicted it and it actually was true, and that means it's sort of a decent scientific hypothesis, or more support for what we already thought was a decent scientific hypothesis. And so, again, the model predicted this was possible. It guided us to the actual specific changes. It isn't just a random change. And looking ahead, it says that, you know what, it didn't matter which image you started me with. For any image, I'm pretty sure I can find some kind of perturbation that will cause a large change. So it's not just I'm telling you, here's this one image to change. Imagine any starting image. We could start with any clean image. And it says not only for any clean image is there only one change, but there's many possible changes that could also produce this effect, which is especially important when we start to think about modulating more than one neuron, that we have many options to try for each neuron, because we may want to do them in combination. In fact, that's what we're trying to do now. So intuitively, I want you to think about the ideas. The models have pointed us to something you might think of as like holes in the visual fabric of the visual system. There are ways to do slight modulations that you might notice are barely perceptible to us, yet can cause large changes deep inside the system that we're studying, in this case, the visual system. Now, you could think of those as defects. Computer vision scientists might say, that's a problem, we need to patch it up. But biology may have not ever had a reason to essentially patch those up, because only with technology where it modeled this well and then finding these kind of interventions would it even be possible. So I think of that as an opportunity, perhaps, to do beneficial things in exactly the sci-fi way that I was telling you a moment ago. And we don't want to just modulate one neuron. We'd like to modulate many neurons. And remember, our models already tell us how to do not just one neuron, but many neurons, and that we actually had showed that already in that earlier paper I referred to.
New slide titled A Speculation, that holes in the neurobiology, exploited in a coordinated way, might one day be used to improve human well-being without disrupting natural visual life. The brain pipeline returns at the bottom with a Yoda image as input.
Okay, so now we're on this sort of quest, if you will, to try to sort of give this idea some legs, some further legs beyond this discovery, to sort of imagine this idea. And the idea is, imagine that you're going to continuously make changes, highly coordinated, subtle changes to the video feed that's striking your retina to produce some beneficial application, change deep in your brain. And so this would be non-invasive, nothing in your head, and maybe, again, without perceptual disruption, if we could. So I'm going to tell you about some beneficial applications that we're thinking about in a moment. Before I do that, I want to first tell you, how could you imagine even delivering such a thing?
New slide titled, Such modulation of one's normal visual input could potentially be delivered via AR glasses without disruption of natural visual life, with photos of AR glasses being held and worn.
And here we think about sort of things like AR glasses technology. So just like many of us wear, I have contacts on now, but many of you wear sort of acuity glasses, you put them on. AR glasses are such that they can capture the images coming at you and potentially change them on the fly so that the world strikes you not exactly as it is, but is slightly different than it is per every image that you choose to look at. So I'm not proposing you go into a room and stare at a bunch of pictures of beaches and puppies. I'm proposing that, hey, you put on your glasses, and there's a slight modulation to every image that you happen to look at in a way that's going to help you in some beneficial way. Could be delivered by AR technology glasses. Okay, let's talk about what do I mean by beneficial? What could we imagine? Why would we want to modulate neurons, and how could that be beneficial?
This is a return to a previous slide, now titled A Speculation, that holes in the neurobiology, exploited in a coordinated way, might one day be used to improve human well-being without disrupting natural visual life. Below, the brain pipeline returns with a Yoda image labeled non-invasive, feeding through stages LGN, V1, V2, V4, and IT. Two callouts read, IDEA, continuously make subtle changes to the natural video feed on your retina and cause highly precise, beneficial neural modulations deep in your brain. A yellow note asks, human beneficial applications?
Well, so there's many possibilities here. You have to think about, for us, we think about the visual system that we're modeling. Where does it connect to? Connects to areas involved in task control, memory, decision, and action. But the one that's most intriguing to me at the moment is an area called the amygdala.
On the slide, 3 brain regions now branch off from the IT stage. Prefrontal cortex labeled task control, peroneal cortex labeled memory, and basal ganglia labeled decision and action.
Which is part of the limbic system, which is involved in things like emotional regulation, and is thought to be disrupted in things like mood disorders or anxiety disorders. So, and often people think about stimulating the amygdala as ways to perhaps improve some of these things. And here we're sort of thinking about the idea of, could we modulate in a way to sort of just tickle the amygdala in just the right way, in some non-invasive way, to do that, potentially to effect sizes that are just as good as we might do with some invasive technology.
Photos appear on the slide showing scenes of emotional distress and counseling, accompanying text reading inappropriate neural states here implicated in anxiety/mood disorders.
Of course, you know there's psychiatrists in 2021 declared a national healthcare emergency among our nation's youth. In fact, many patients are seeking non-invasive, non-pharmacological treatments. So the time to try to explore things like this, we think, is right.
A detailed inset appears showing the amygdala broken down further, labeled amygdala BLA, containing a positive valence neuron and a negative valence neuron, with other inputs feeding both.
Think about the amygdala now, just to give you a very simple model of the amygdala, totally oversimplified. This downstream brain area that IT cortex connects to directly has neurons that tend to activate when something good is happening, called positive neurons, and positive valence neurons, and other neurons that activate when something bad is happening, negative valence neurons, carried by other inputs from the brain. And so the visual input is implied by IT, so we can now think about modulating just the positive valence neurons up, for instance, to say might lift mood or reduce anxiety. What that amounts to in us, in a geometric sense of what we're trying to do in our models, is to think about an area like IT that's projecting to the amygdala, and this might be the state of activity across all the neurons in IT caused by this image.
On the slide, a new diagram. On the left, photos from a street scene labeled pixel space. On the right, a 3D coordinate system labeled IT neural population state space, with axes for activity of neuron 1 and neuron 2. Black dots represent neural states from each fixation, connected by zigzag lines showing eye movements.
And when you move your eyes around and explore that scene like the one I showed you at the beginning, each time you move your eyes, you get a different set of photons on your eyes that then produces a different state in your IT. And the arrows here are just indicating your eye movements, but what's causally driving these neural states are the photons pushing through your ventral stream to activate those neurons. And now what we're going to try to ask the model to do is do something consistent here, where we make a little bit of change to every image, contingent on the image.
A green callout reads, "Make light injections. Adjust each incoming image in a way specific to that image." On the diagram, green up arrows now lift each neural state point upward.
So that we might move the IT state along a particular direction. In this case, I'm drawing it up. You can think of that as like, let's turn on one IT neuron a little more than it's always activated, and all the other ones keep doing what they were doing. That's the simple frame, but the general frame is we can push in any direction in the population, but that's the sort of way to think about this. And we might call that lifting the population along a particular direction. And if this green line is, for instance, aligned with a direction of the amygdala neurons preferring, that would be the sort of application version that we're dreaming of. For now, we're just testing it in IT itself. And when I say we, it's led by Guy Gaziv, who was a postdoc in my group and is currently at Meta.
Photos of the team appear. Guy Gaziv, Sarah Goulding, Yoon Bai, and Ani Avazian Hancock.
And this is the team that did the work that I'm just going to briefly show you here.
The slide shows a heat map labeled Experimenter Chosen Target IT Neural Population Lift Patterns, and the pattern of activated neurons visibly spells out the letters MIT.
So the idea is like, what if we could inject various patterns? We're not just going to lift one IT neuron, but just think about injecting, turning on this neuron, turning off this neuron, activating these neurons and not this one. Here's like 6 neurons here. So this is one direction, one of those green arrows, for example, here. And we tried a whole bunch of them here that we made an acute pattern for reasons that I hope are obvious to you. So here's an example of what we actually found in the monkey data when we recorded a population of neurons trying to do this. And you can see that we can essentially lift these patterns, in this case, a pattern of our choosing that we just happen to like.
A second heat map appears to the right labeled Observed Results. It shows the actual recorded neural responses, which roughly reproduce the MIT-shaped pattern. Slightly noisier, but recognizable.
And we could lift up the neurons, and this is the sort of data from one animal. You can see it's not perfect there, but you can see that we're able to be pretty precise in how we're able to tune our image modulations to change neurons in whatever way that we like here, as just these examples show.
A photo on the slide shows the Charles River outside MIT with numbered yellow boxes overlaid showing successive fixation points.
We did this also not just in kind of laboratory conditions. We did it where we sampled what might be a natural feed of, say, you or I looking around a scene like this one of the Charles River outside MIT. And so that suggests that we could do this in that kind of AR glasses style in principle. We still don't know if this would have strong clinical effects or if we can tune this to the amygdala. And so that's really why this is still a science fiction story. We're pointing towards the amygdala, but our experiments have been in IT so far.
New slide, a visual showing two head profiles facing each other. On the left, the visual system glowing red at the back of the brain. On the right, the limbic system with the amygdala highlighted. A green arrow connects from the visual system to the amygdala. Caption: this story is still science fiction.
We don't know if we can, in some sense, push enough neural energy through to cause this to produce meaningful human health benefits, but we are intrigued by the possibility that we could, and we're doing ongoing experiments now to test this idea. So now what we're trying to figure out is the path from IT to the amygdala. What are these native connections here if we want to tune this along this axis here? And that's what we're trying to model at the moment to extend our models there. And when I say we, I mean Alina Peter and Guangzhou Kim.
A new banner reads, "We are now determining which specific IT population modulation patterns should up or down regulate amygdala valence neurons." Below, a panel labeled Example Monkey Amygdala Visually Driven Neuron shows 6 raster plots paired with different stimulus images, vegetables, fruits, jungle scenes, and rope textures, illustrating that this neuron responds differently to different categories.
Who are busy recording from the amygdala and building models of that. What I can tell you early days is our models readily extend to those neurons. These are now these kind of positive valence neurons. And I can also say that we have evidence that that positive valence neurons have a different visual preference drive than negative valence neurons, so that suggests this is possibly to be true even without any training of the monkey. Okay, I'm going to zoom out now. That was a science fiction story. It's still in progress. I told you 2 stories today. The first story I told you, and I want you to take some takeaways from, the first story is I told you a story of how a subset of perceptual states, the states of is it a car, is it a dog, is it a person, those mental states, how they emerge from brain mechanisms and our latest computer models of that, where they came from and how they continue to evolve. The main takeaway there is that these multi-scale models that cross from neurons to networks of neurons, they've been discovered via AI-forward strategies. That doesn't mean AI on its own, but AI coupled with neuroscience and cognitive science to continue to run that virtuous cycle that I described. We think this is going to continue and expand, not just in vision, but other areas, some of which I alluded to. The second story I told you was a possible science fiction future. Why should anybody care as we build these digital copies of parts of our brain function? What can we do with them? And I gave you sort of one example of how we might modulate mental states. This is just one example of myriad applications that might emerge. It was just one that was, to us, one that we thought we could pursue near term. So this could, imagine in the long run, unlocking all kinds of new technologies to improve human health, which I referred to, but also things like human education.
This is a return to a previous slide labeled AI-engineered systems as multi-scale scientific models, shown as 5 linked stages: neurons and synapses, neuronal circuits, brain regions, states and algorithms, and behavior and cognition. A new 6th box is added to the front of the pipeline: molecules and genes, extending the multi-scale model one level deeper.
I want to make a nod to the fact that these models right now, they go from neurons up to behavior and cognition, but of course these neurons are maintained and activated, controlled by a whole bunch of molecules and proteins that control their activation state and how they even work at all, maintaining them as functional processors. And so as our molecular colleagues start to build models of how we can connect those molecular tools into the activity states of neurons, that unlocks a whole other kind of interventional toolbox beyond showing light on the eyes, but being able to predict, for instance, how CRISPR gene modulations might full stack produce behavioral and cognition states, if that's even possible, which is really a long-standing full stack dream of the field. So the models at some point will connect to that level as well, and that will open up an entire toolbox even beyond things like AR glasses and the kinds of things I showed you today. So I want to just say here in closing that this idea that I started with that's a bit provocative, again, something like 95% of the population believes that there's something more to our brain function than just a machine underlying that. That as we build these models, that will change our viewpoint on ourselves, that more and more of the population will start to see, actually, we are machines in this way, and that will be potentially disruptive. But we've lived through these disruptions before, these 2 major ones. We used to not think that we were involved from organisms like monkeys. Now we largely accept that. We used to think we were the center of the universe, right? And we now don't think that anymore either. And those changes were disruptive at their times, but we survived them, and I would say are even better for them. And I think the same thing is going to happen here as we start to accept these notions of ourselves. And I wouldn't say we're just merely machines, but we are amazing machines. And as we understand those machines, look at all the things that we can do and fix and do better than we couldn't have done before we knew that. And so as we understand a sort of science of intelligence, a science of understanding ourselves, reverse engineering the kind of machine that's essentially running inside all of our heads, we'll of course have things like impacts on AI and the back and forth between AI and brain sciences. But also new ways to think about teaching our kids and ourselves, new ways to maybe intervene in mental health conditions, of which I gave you an example.
Final slide titled, Our Goal, Understand Intelligence, with 4 application areas surrounding a central head and circuit board image. Aligned AI systems improving the world, recognizing cultural influences to avoid conflict, deep knowledge of learning to teach all humans, and engineering-level understanding of mental health.
And maybe new ways at the societal level of how we interact with each other to do things like avoid cultural conflict, as we build models of how we interact with each other at those bigger scales. And I think I'm very bullish on seeing this happen to our species. This is a time that it's beginning to happen. I hope I've given you an example of that. And if you like these kind of things, here's a few examples of papers that you can go and read more about it. And I'm really, really thankful for the time that you guys have given me to listen to me, and to the organizers for inviting me to even give this talk. With that, I'll say thank you. So I have, there's questions, if anybody wants to ask questions. There is a mic in the room, and there is about 5 minutes of time if people would like to ask questions. Or you could try to yell if you don't have to get down to the mic. Yeah.
So, a really good population for the IT neurons. Okay, so we typically think of IT neurons as monkey lymph
As
monkey lymph. They're the monkey lymph nodes.
the monkey what?
Things like monkey lymphs or pictures of
Oh, I see. So you think of like things like faces or other things like that.
yeah, faces or lips or whatever, like these kind of higher level conjunctions of features.
Mm-hmm.
But then you showed your modulation was much more effective in driving that neuron than the typical monkey lips that you expect in IT. So does this imply that with better ways of mapping neural pathways, we'll have a different understanding? Like this is our new chronology.
Yes, well, I think already, I was never a believer. Humans would like to look at the neurons and sort of, say, oh, I think that's the neurons or faces. And of course, there is a preference for some neurons preferring faces and lips. But I always assumed it was going to be a little more complicated than that. In fact, a lot more complicated than that. And the one thing you can think of, the models, they already gave us a set of a vocabulary, if you will. You could call it, I don't know, you might have said a phrenology, but it's sort of a list of nonlinear functions, I would say, that are a better descriptor of what the neurons are doing than words like faces or lips or whatever, right? So the models gave us that, and they're still giving us that, but that vocabulary is still being worked out. But then, yes, to your point, that becomes our operating space to say, that vocabulary is not quite right, we need to tune it up a bit, and that's where that's headed. But yes, the prediction to modulate the neurons essentially comes from, if you adopt this vocabulary, it can also then tell you what to do to cause things like activations. And again, that was just one example neuron to just sort of highlight the ideas. Of course, all the neurons we see do all similar things. So it's not special to that neuron or the fact that I was showing you a picture of a dog. But yes, many of the modulations live in spaces that look nothing like things that are semantic. And I find that quite important and potentially interesting. And it relates to the idea that I mentioned about how can we do this without modulating perception. There may be a kind of perceptually null space that we can work in there, we hope, where the models tell us, this is going to modulate the neuron, but we are perceiving faces and lips, and it has no impact on the things that matter to us, like faces and lips. And that would actually be what you'd want. If you take a drug like Prozac, and then when you don't want to walk around thinking that you took it, you just want to live your life, but this is happening in the background. So if there's room in there and that sort of degeneracy in the IT code with respect to perception, then we could maybe leverage that. But we don't know that yet, but we believe there might be. Those examples hint at it, but remember, we're only controlling a few neurons at a time, so we don't know if the relationship between perceptual states and IT codes is unique, or if there's some degeneracy that we can leverage in there. Sorry, your question gave me a chance to sort of add that bit into my talk, but I hope I answered your thought. Okay. Yes?
So
Yeah, to the extent that the number of neurons and their connectivity in the model and in the biology are disparate, how do you measure the environment?
Okay, so you're asking about you're asking first, you're sort of stating that the anatomical connectivity in the biological network might be a bit different than the connectivity in the artificial neural network. In fact, I don't know offhand the best study on how to say how far off it actually is. I mean, it's loosely inspired, but it's not exact. The comparisons that we're making are at the functional level of the units of the neurons themselves, a little bit ignoring that connectivity. As the connectivity is more aligned, you would naturally expect, as the anatomical connectivity gets more aligned, we would expect the functional connectivity to improve. I hope I said many times that these models are not perfect. They're not perfect, but they're already useful as a way to think about them, right? So as we do things like connectomes, and that's happening in flies and maybe eventually in humans and monkeys, then we could get that exact connectivity, and that would produce even more veridical functional alignment. The point I'd like you to take here is we already have very strong functional alignment, even if we've kind of not guessed the anatomy quite right. So I take your point that there's more work to be done there in terms of making it better. That's the metric we've been using. And in some sense, you could think of it as, in some sense, the behavior is, in some sense, the ultimate metric. If I want to influence your behavior, maybe your anatomy you'd like me to change too, but if I change your anatomy, my colleague likes to say, hey, people don't care if you have plaques and tangles in your head. They care that you're losing your memory in Alzheimer's, right? So it's like the functional level of behavior is what matters to the human species. So that gives it a privilege level, but of course the anatomy relates to that. So all the metrics I've showed you were largely functional. We don't use a lot of anatomical metrics yet. But as I mentioned, that's coming online for simpler species, so-called connectomes, where we can get much more, in some sense, it's called animating the connectome. They build the connectome and they go, what's it doing? And they have to try to simulate it and start to run up. Don't yet have those data in monkeys, but the field has it now in flies and is going to progress that way. So that will be even more advanced models that are going to better align to some of the molecular tools I mentioned too that we're able to do with these kinds of models. That's sort of on the horizon in this decade. I think maybe one more quick, and then I should go because it's about 5. So how about we do that?
2 maybe very questions.
Oh, 2, okay.
If you subtract the original dog from the modified dog and take a high contrast image of that difference, does that stimulate the same neuron?
It's a good question. We didn't do that experiment. It's a pretty small change. I doubt it will, but we didn't try that, so I can't say for sure. I don't know if it has anything to do with that in particular. The question is but I think there's something important here about thinking what is the native kinds of connections into our amygdala? What are we all naturally, visually, you might say, afraid of or pleasing to each of us? The amygdala is often thought of an area to learn with how to associate any visual thing arbitrarily with good or bad, but we all probably have some native connection which might relate to our fears, which might be overblown in some people and less than others. And when I mention those amygdala studies, we're trying to understand what are those native patterns without any training at all. And that's going to vary by individual, but we think there might be some shared, both primate and human shared space that we can leverage here. But an idiosyncratic model of each of us would, of course, be far more powerful, for example. But that's in the future, but it's a great question. I'm going to stop there and say thank you. Yeah. Sorry?
Jim, let us do a bonus. Can you leave the last question to either undergraduate student or graduate student? You have a chance to ask a question. Make sure you are a graduate student or undergrad student. Undergrad, go ahead.
Perfect, yeah. This is a great question. So the question is, and it's sort of all technologies come with potential downsides, right, as you're sort of doing these kind of research, right? So it's something that we're actively working on. We have something called the social and ethical responsibilities of computing at MIT, and we're working with them to come up with ways that we can try to think about regulating this technology. It is still a work in progress, I'll admit, but it's something we think about as well, like how we would not want this to be developed and used for negative things. Although we have to also expect that most technologies, opioid crisis, for example, opioids are great for medicine, but then they have some downsides. So there are going to be risks associated with any technology, but it's important for us to be thinking about it exactly the ways that you say. And if you're interested in that and have ways to help us do that, would love to talk to you some more, because I think it is one of the challenges. It's also true beyond of all AI methods, not just models. Here we see using this for beneficial, but it's a general challenge, deep fakes, all of the things that kind of technology brings. So I'm glad you're asking that, and I don't have an answer to say exactly how we're going to do it, except to say we are working on it, and we're coupled with postdoc fellows that are working on that right now. It's something that we are actively trying to figure out. But I'd love to talk to you more after if you'd like. Yeah. Thank you. Great question. Yeah.
Okay, it's again a great honor to have you here, and elegant work, and also beautiful insight. Let us thank Jim again.
Closing slide reads, Stony Brook University, State University of New York, stonybrook.edu.